everything by making
Mertens.
resting stages.
Okay, thank you, Pascal, and thank you very much for inviting me, and thanks for coming to listen to this talk. Okay. So I work for Ifremer. For those of you who do not know Ifremer is the largest oceanographic institute in France. So it's really a big institute. We're like 1,500 people, and we have 5 large centers, but we also have a lot of small sites. We have 26 sites spread all around France, and so there's many places all along the French coastline. But we also have labs in the colonies of France, like French Polynesia, Réunion Island. And we have a very large fleet for sampling. So why Ifremer is interested in microalgae? Actually, Ifremer wants to ensure human safety by monitoring toxic microalgae that can cause intoxication of shellfish or fish gills. So back in the 80s, in 1983, there was a major intoxication caused by dinoflagellate Dinoflagellates acuminata in Bretagne, and so there was over 3,000 cases of diuretic shellfish poisoning. And after this, they decided to erect the plankton monitoring network, which was the REFI plankton monitoring network, which was erected in 1984. So I work in a small town called Concarneau, which is in the south of Bretagne. It's here, and so my team, the people I work with mostly are work on the morpho-molecular taxonomy of microalgae. We frequently work with our colleagues from Nantes who are specialized in toxins, and our colleagues from Brest who do more molecular ecology of microalgae. So what are dinoflagellates? Dinoflagellates is a group of microalgae that sometimes can be toxic. So there's about 2,377 extant species known based on morphological data, and the majority is marine to brackish, and the majority is as well planktonic. Something that dinoflagellates sometimes do is that they can produce cysts as part of their life cycle. These cysts are produced during the sexual phase, and these sink into the sediments, stay there for a certain amount of time, which can be a couple of days or several years, depends on the species. And after this period, they hatch and they restart the cycle. In 2015, there was a very important paper in Science that looked at plankton diversity, and this was led by a team from Roscoff in Brittany, and in this paper they looked at samples that were took by the Tara schooner, who sailed all around the world and took samples at many places. They took back these samples to the lab and did metabarcoding on the samples, and so they tried to assess the diversity using this metabarcoding approach. And in this paper, they show that the biodiversity derived from the operational taxonomic units, the OTUs, is actually much higher than the known morphological biodiversities. So, for instance, for dinoflagellates, it's shown here. This blue bar is the OTU-based biodiversity, and the red one is the morphological based. So they said, using this approach, we can kind of show that there's a large biodiversity currently unknown for dinoflagellates and other groups of organisms. Today I'm going to talk to you about a particular group of dinoflagellates that we are working on in Ifremer, particularly because my colleague Elisabeth Nezan has been seeing more of them in recent years in the plankton observations that we do at Concarneau, and we want to try to understand why we see more and more of this species. And I'm going to specifically talk about this group because it's a fairly unknown group, and it's a good example of what we don't really know about diversity of dinoflagellates. So this group of dinoflagellates is called the Podalampadaceans, and there's 6 genera currently described and 35 species. So these dinoflagellates have a specific tabulation. For us, it's easier than for flat forms. We can just look at the plates, and these plates help us to identify the genus. And then if we go to species level, we'll look at smaller details in the morphology. So we looked at samples from Concarneau and from the Mediterranean, also from the Union Islands. And we also had samples from in the North Atlantic from a sailing boat that took several samples for us. We used light microscopy and scanning electron microscopy, and we did single-cell PCR on the cells. This is necessary because we cannot really grow them. They're heterotrophic. They eat cyanobacteria, so they're very difficult to culture. And we use a method that allows us to obtain a long sequences up to 4,000 base pairs, which include SSU, ITS, and LSU. So as a result, from the morphological observations, we found that we could identify 18 species that belong to 5 genera. The 6th genera we did not find, but it's a very rare genera and genus, and it's only been observed one time in the Indian Ocean, so it was not very surprising. So just to show you the diversity within this group of dinoflagellates, this is the one of the genera, Blepharocysta. So you see the cells look very similar, but when you start looking at the details, you start seeing the differences between the cells. These are the 3 different species. These are 2 more, so you see quite a big difference in size. These are 2 undescribed species that we found. Under the 2nd genus, Gaarderiella, we found 2 out of 4 described species. For Litsodinium, we found 4 out of 18 described species. And for Mysticella, we found 1 new species. And the more easy to recognize, the genus that's most easy to recognize is Podolampas, and here we found 4 out of 5 species. For the molecular work, we had it's important to note that we had a very low success rate, so we don't really have 100% success rate, but it's difficult because we had lots of problems with contaminants or mixed sequences, and this could be due to presence of fungi or endosymbiont or prey. So we obtained 30 new sequences of SSU, rDNA, and this allowed us to identify 22 species and 5 genera. And so this is really a big step forward because there was only 5 sequences currently in GenBank of SSU. And so the phylogenies show very well supported family of the Podalampadaceans. Also on generic level, the SSU phylogeny was very useful. But on species level, it didn't really work very well. So it was necessary to look at another gene. So We also looked at the LSU, and the LSU phylogeny showed that that we could really separate the species very well. It was necessary to transfer two species of Blephariocista to a new genus because there's differences in the tabulation, and they are in different clades and the phylogenies. So we're going to erect two new genera to describe these two species. So what's important in terms of biodiversity is that so we looked at part of the SSU to the small subunit DNA to see whether these small parts can be used to identify all the different species that we identified using the whole genomes or in combination with the morphologies. And actually we did this because the approach that was followed by the people from Tara Ocean expedition was actually they looked at the V9, so only 130 base pairs to try to estimate the biodiversities. But when we do this for our Podolampadaceans, we find that we could only define 7 non-identical groups. We could not discriminate 22 species. So it's a bit difficult to understand how these operational taxonomic units that they use in Tara Oceans, how they translate that to biodiversities. So in conclusion, we found 30 new sequences of and 37 LSU sequences. We found Blephariocista species and Ishicella, and we found good support for the genus concepts for LSU, but species concept was necessary to be supported by LSU. And so we could not discriminate V4 or V9 of SSU. So it's important to note that high-resolution morphomolecular studies are still indispensable today for biodiversity estimates. I think Tom already showed that very well. But we have a very big problem there because we can only do so little in so little time, and it's very difficult to bridge the gap between how many species are out there and how little people there are to do the taxonomic work. So, but thank you very much.